In this video, you will learn about chemical communication in the autonomic nervous system. Let's start by looking at the neurons that release the neurotransmitters acetylcholine and norepinephrine. Cholinergic fibers are axons that release acetylcholine. You'll find cholinergic fibers in several places. All parasympathetic fibers and all preganglionic fibers are cholinergic. Also, some of the sympathetic postganglionic fibers are cholinergic, including axons that innervate sweat glands and arrector pili muscles. Adrenergic fibers are axons that release norepinephrine. Most postganglionic fibers in the sympathetic division fall into this category. So when you see adrenergic, think sympathetic activation, fight or flight. Now, the effect of these neurotransmitters depends on the type of receptor they bind to. The same neurotransmitter can be excitatory or inhibitory depending on that receptor. Let's look at cholinergic receptors first. These are stimulated by acetylcholine. Nicotinic receptors are found on all neurons in autonomic ganglia and the endocrine cells of the adrenal medulla. When acetylcholine binds to nicotinic receptors, the effect is always excitatory. Muscarinic receptors are found on all effectors that are regulated by postganglionic cholinergic fibers. This includes the target cells regulated by the parasympathetic division. The effect here can be either inhibitory or excitatory, depending on the receptor subtype. For example, acetylcholine has an inhibitory effect in the heart, slowing down the heart rate, but it has an excitatory effect in the digestive organs, increasing activity of the smooth muscle and exocrine glands. Adrenergic receptors bind to norepinephrine and epinephrine, while norepinephrine is the primary neurotransmitter released from postganglionic fibers in the sympathetic division. Both epinephrine and norepinephrine are hormones produced by the adrenal medulla under control of the sympathetic division. These hormones travel in the blood to amplify the fight or flight response throughout the body. Now that we understand the basic chemical signals used by the autonomic nervous system, let's look at how certain drugs can affect these systems. Many drugs are designed to either mimic or block neurotransmitter effects, creating either excitatory or inhibitory outcomes on target organs. Let's start with two key terms. An agonist is a chemical that stimulates a receptor, mimicking the natural neurotransmitter. An antagonist inhibits a receptor, preventing the natural neurotransmitter from binding. Nicotinic agonists are drugs that stimulate nicotinic receptors in both autonomic ganglia and skeletal muscle. Because both divisions are activated, their effects can cancel out in many organs, though sympathetic effects are often dominant. Nicotine and succinylcholine can cause paralysis by disrupting neuromuscular receptors, but they also increase blood pressure by stimulating sympathetic responses. We can also categorize drugs based on which division they affect. Sympatholytic drugs are adrenergic antagonists that block sympathetic effects. A common example is propranolol, a beta blocker that decreases heart rate. Sympathomimetic drugs are adrenergic agonists that stimulate sympathetic effects. Examples include albuterol, dobutamine, and phenylephrine. Albuterol is commonly used in nebulizers and inhalers to dilate the bronchioles as treatment for asthma. Dobutamine is commonly used to increase cardiac output. Phenylephrine is commonly administered by ophthalmologists in the form of eye drops that function to dilate the pupils before examination of the retina and other internal structures of the eye. Parasympathomimetic drugs are muscarinic agonists that stimulate parasympathetic effects. Pilocarpine is an example used clinically to constrict pupils, reversing the effects of phenylephrine. Parasympatholytic drugs are muscarinic antagonists that block parasympathetic effects. The classic example is atropine, which increases heart rate by inhibiting parasympathetic stimulation. Atropine was first discovered in plants of the nightshade family. In summary, cholinergic fibers release acetylcholine, which binds to nicotinic and muscarinic receptors. Adrenergic fibers release norepinephrine, which binds to adrenergic receptors. The receptor subtype determines whether the effect of a neurotransmitter is excitatory or inhibitory. Drugs can either mimic or block these effects, allowing clinicians to regulate autonomic functions. Understanding these interactions is key to both physiology and pharmacology. It helps us predict how the body will respond under different chemical influences.